So today I'm going to talk a bit about risk prediction in COVID. As Sean said, a lot of my career has been looking at methodological challenges in causal inference. But when COVID came along, obviously things changed for an awful lot of people. And one of the things that I got involved in was the question of who's at highest risk of COVID. And today I'm going to share some thoughts that came up as part of that process. So I'm first going to talk a little bit about the OpenSAFELY collaborative, which is the platform with the data that I used to think about these questions, and then talk a bit about some of the challenges that we encountered. And in the latter part of the talk, I'm going to talk a bit about the idea of modelling absolute risks in this context. So OpenSAFELY is a new collaborative that was initiated in response to the COVID pandemic. And it's a collaboration between the Oxford Data Lab, which is now the Bennett Institute, and the Electronic Health Records Group at the London School of Hygiene and Tropical Medicine, where I work, and TPP. And TPP is a vendor of electronic health records. So in their day-to-day business, they hold the electronic health records of a large number of people. So I've said virtually all of that. So now this slide describes OpenSAFELY in the early days. Right at the beginning, it held data for 40% of the people in England. It's now expanded beyond that. And OpenSAFELY is built on a couple of really key principles. The main one being protection of patient privacy. And it's slightly different from many other similar sorts of data resources in the sense that with OpenSAFELY, the analyst never gets to see the data. So the internal software uses these tiered, increasingly non-disclosive tables, and all this analysis that you're going to see was done without me ever seeing the actual underlying data. So what's in this data set? As I said, it's now beyond just TPP, but at the time I'm going to talk about, we had primary care electronic health record data for 24 million people. And that was linked to COVID test results, the CPNS, death data, and intensive care data. And I understand that's more than 10 terabytes of data. And I think that equated to more than 60 billion rows of data. So this is huge. And just to give you a flavour of how OpenSAFELY works, if you haven't encountered it, all analyses run using OpenSAFELY data or the data within the platform. There's pre-registered protocols, and there's an open tool for code lists. So all code lists that are used in an OpenSAFELY project are available for reuse, and it's searchable with version control, and you can find that online. It's called Open Code Lists. So that's a really nice resource for anyone analysing EHR. And so you create your data management tool script, which tells the system what variables you want and how to define them. And then you push that to the system and it generates fake data. So a dummy data set, which has the same variable names and the formats that you would get in the real data, but it's dummy data, it's not real. And then you develop your analysis script on that dummy data. And when you're confident that it works, you push it to the system and it's run against the real data. And then you can download redacted results. So it's just an interesting part of the way that it works, that you never get to see the underlying data. So it was a real challenge learning how to analyze data without actually seeing it. Now this is the first thing we did with these data, was to explore who was dying from COVID in the very early days. And you can see on the right hand side, this paper was published 3 months into the pandemic. And this is not to show off how amazing our team were, although I think some of the team listed there are truly amazing. The point is that these data, because they're routinely collected, you can do research in a really timely manner. And so they're an incredible, powerful and timely resource for research. So risk prediction, what are the challenges? Well, I'm sure this isn't all of them, but let me tell you about some. So as we all know, we've just had a pandemic, and early on there was an urgent need to understand who was at highest risk. And why did we care? Well, firstly, for risk minimisation, the government had to know who to advise to shield and what sort of work at home guidance was needed. And as the vaccines were developed and available for use, who should be prioritised to get those vaccines first? And more recently, we've got the neutralising monoclonal antibodies and antivirals. And again, these need to be prioritised. And all these questions require some idea of who is at highest risk of the most severe outcomes. So I've sort of grouped the main challenges that we encountered into three main sets. So one being, who's the target population? One being sort of more nebulous, how do we formulate the whole problem and what sort of predictors do we include? And the third about this particular context where the underlying risk changes quite drastically over time and the consequences of that. And I'm going to spend quite a bit of time thinking about that last issue. So the population. Now, we could think about various populations who are of interest for this question. Do we want to know the risk of dying from COVID just for the general population, or the risk among people who have been exposed to COVID, or the risk among people who are already infected? And those are three perfectly valid but different questions. And we might well find that different groups of people are at highest risk of those different things. So, well, what can we answer in routine data? Well, general population, we don't have the whole population, obviously. There are certain groups who are excluded largely from electronic health records, like people who are homeless are typically fairly excluded. But we've got a fairly good grasp on the general population using electronic health record data. So we can at least make a good stab at trying to estimate risks in that group. Is it relevant to policy? Well, I think it is. Early on in the pandemic, we provided PPE to medical practitioners, not because we thought they were particularly high risk once infected, but because their overall risk through those different stages was high because of their higher exposure. So you can argue that the risk in the general population is of policy interest, and it can be targeted more or less in these sorts of EHR data. Obviously, we've got no sense of who's exposed. Can we get at the population who are infected? Well, you could argue that if we have widespread routine testing and that test data is linked, then potentially we could get at that population. Have we ever had that? No. So in the early days, testing was very scarce. And even when testing was very available, people who did tests and then people who logged results of tests weren't a random sample of people who were infected. So it's very hard to get a handle on that group in these sorts of data. So really, if you want to use electronic health record data, there are restrictions on the population that you can explore. And obviously that brings complexities into how we interpret the results. Okay, so the question we're trying to answer here is who's at highest risk of COVID mortality? And this rather ambiguous title, Who is the Who, is about what sort of answer we want. What format do we want the answer to come in? Are we after a list of people who are at high risk? Or are we after a list of conditions that place people at high risk? Or a list of patient groups identified by a condition who are at high risk? And that might sound quite like the previous, but it's not, in the sense that learning disability for interest, for example, in itself doesn't or may not increase risk very much, but because people with learning disability tend to have all sorts of other characteristics and comorbidities, when you think about the set of all those characteristics together, that particular patient group is at higher risk. So there's a slight difference between the conditions and the patient groups who have a condition. Now, what can we easily provide? Well, statistical methods and methodology that exists is largely targeted at providing a list of individuals at high risk. So risk prediction models. We can provide lists of individual conditions that have higher observed risk just through basic descriptions. Now, the list of individuals is very helpful if that's how the policy is going to be implemented. If, you know, people are going to come into a GP and the GP record has a risk score, and that's when things happen. But particularly in the initial parts of the pandemic, things were being implemented more broadly, and the sorts of policy categories they wanted were much simpler. So they were more interested in having a set of A, B, and C, because you're higher risk. And that's not quite what our risk prediction models are set up to do. So in practice, what happened? Well, the early list of high-risk conditions were largely based on clinical expertise and existing lists related to influenza. And then as research began to be done in this area, often people would pick out coefficients from risk prediction models to select the conditions with large coefficients. And it's simple, it's very transparent, but is it doing what we want it to do? Well, just to very briefly have a think about that, on the left-hand side, we have the absolute risks from a multivariable model, where these are the absolute risks associated with having just one of the different comorbidities or characteristics and nothing else. And there's 5 colours here representing the different categories of risks. So we've got light green indicating the lowest risk, and then dark green, so mustardy, greeny orange, and then red are the highest risk group. And if you just think about people having one of those characteristics at a time, none of these characteristics put people in that really high risk category. Whereas if you look at the average risk predicted for people with that condition on the right-hand side, suddenly we're seeing that particularly people with kidney failure and organ transplant are in that very high risk group. So people with certain comorbidities tend to have other characteristics coming along with that. So just picking out coefficients may or may not give us a set of people that we're after. In terms of predictor selection in electronic health record data, one of the biggest problems is missing or absent data. So it's very difficult to measure certain things. Early pregnancy, there's often very little record of it until further down the line. Occupation, there's very little data about people's occupation, and obviously something like an infectious disease, occupation might be really quite relevant. Even assuming that you've got all the data that you want, there's a lot of data. You could have hundreds of potential predictors. So you're stuck with a variable selection problem with rare outcomes. And furthermore, you often find interactions, particularly in COVID, age is obviously the strongest predictor. And if you have a look for interactions with age, you often find sub-multiplicative interactions, and that's fairly common. So if you want to allow for all those sorts of interactions, you've got even more variables. So you've got this sort of big variable selection problem. And in parallel, if policy is what you're interested in, there's perhaps a concern that you want your answers to be clinically sensible. You want some sort of face validity. So early on, people who were looking at risk prediction model had discussions about, well, maybe we should omit smoking from contention, because if we put it in, it seems to be having a coefficient in the protective direction. And, you know, what people then say, oh, I must smoke to reduce my risk and take it in a sort of causal manner. And that, you know, policy-wise, that's probably not a sensible policy. So that there is a sort of vague unease that you want your model to have face validity. And how do we ensure that that happens? And obviously from a policy perspective, we want a risk stratification that can be easily implemented. So if it involves data that is incredibly hard to get for the person that you want to give advice on their risk for, then that's not ideal. So there's an awful lot to think about what should go in your model. And just thinking about the variable selection problem as a purely statistical problem, here I'm going to tell you a bit more about the actual data later, but just for the moment, this is just a model with predicting COVID-19 mortality with lots of different groups of predictors. So we have demographic in the sort of whitey colour at the top, cardiac variables in the yellow, respiratory in the green, neurological in blue, kidney in red, then cancer, immunosuppression and other. And each column represents a different geographical subset or a time subset. So these are just sort of taking extracts from the data in various different ways and seeing whether the variables that are selected are different. And you can see immediately that there is a lot of variability. You know, some things are consistent. So all models pick out some demographics and some cardiovascular things and some respiratory things, but which ones are very different depending on quite small perturbations of the data. And if we're going to then use that model to pick out coefficients that are picked up and are big, then this sort of instability is quite concerning. Now, if this was a causal analysis, I'd be talking about confounding, but it's not. We're thinking about risk prediction, but there is still a sort of distortion that can occur. So if we don't have information on a particular characteristic that's strongly associated with risk, then we can end up overstating or understating risk for patient groups. And things for which that is particularly relevant are things like occupation, household composition, pregnancy, HIV status, which can be quite badly recorded. So there is an element of not quite having the variables that you would like to have in electronic health record data. Now, another thing that happened quite early on was lists of high-risk people were created, and then individual patient groups were petitioning the government saying, well, why aren't we on the list? We want to be on the shielding list. We want to be on the vaccine priority list. And why weren't they on the list? Well, a lot of the time because their coefficient hadn't come up in anyone's risk prediction model. And why weren't they if they really were at high risk? Well, in some cases, because that information simply isn't available or wasn't thought to be included in the modelling. Sometimes because the higher risk experienced by that patient group flows through lots of characteristics so that together it's sort of all jumbled up and it's not clear that that patient group is at higher risk when you just look at the individual coefficients. There might be a very poorly specific measure in electronic health record data. So we did some work looking specifically at learning disability, and there's no good measure of that in electronic health record data. What we do have is a measure that we think is highly specific but has low sensitivity. So in this case, we weren't too worried, but in some other cases, you might have a measure that has very low specificity, and that can understate the risk if that group is really high risk. And obviously there's issues to do with, have you got the model specification right and got all the interactions and such like. Now, the thing that bothered us the most was the quantification of risk. So obviously we have a highly dynamic situation where the underlying baseline risk is changing very rapidly. And so if you take a model and fit it to a certain time period of data, it seems unlikely that absolute risk predictions are going to transport well to the following month or the following 6 months. However, in policy terms, we're often told you should use absolute risks for policy. So the question is, should we be trying to target absolute risks that are transferable, or just give up and say, well, it's not really possible in this situation? So options are, we can try to directly get reasonable models of baseline risk that are transportable over time, which is what I'm going to talk about in a minute. Or we could just accept that things change and fit a risk prediction model and just regularly refit it and recalibrate as times change. And also predicting over longer periods of time mean that some of the ups and downs sort of smooth out longer term. Right, so I'm going to talk a bit about how to estimate absolute risks by directly modelling the risk. Okay, so clinical prediction models are typically set up assuming that the underlying context remains relatively stable. And for COVID-19, that's obviously not the case. The burden of infection changes drastically over time. And what's the impact of that going to be in classic risk prediction models? Well, probably in poor calibration, so poor agreement between predicted and observed risks. Do we expect it to have effects on the discrimination, the ability to discriminate between those who do and don't have the outcome, or the ability to rank people in terms of their risk? That's very unclear. So we decided to try explicitly incorporating measures of the burden of infection into our risk prediction model to see if we can get transportable estimates of absolute risk. So we started with a fairly simple model for the hazard of COVID-19 death on a particular day. And this is comprised of 3 components. So in order to die from COVID-19, one has to be in contact with someone who's infected or be in contact with the infection in some way, acquire the infection, given contact with it, and then die today, given that you were infected so many days back. And this assumes that that first infection occurred between little m and big M days ago. And those are just numbers that we sort of just pre-specified for the analysis. Now, we don't have time to go through all the assumptions, but under a number of assumptions about the form of each of those components, we can write that hazard or rate as a constant, not depending on time, times a factor here that we're thinking about as being the effects of individual patient characteristics. Now, obviously, this is not a causal model. We're not thinking about effects in any causal manner, but these are the contributions of patient characteristics to that risk. And then these etas are the probabilities of dying today, given infection so many days ago. And you can see that there's one of those for each of those days where we're considering that infection that many days ago may have led to death today. And then we have this F term. So this term combines the prevalence of infection, the proportion of the population who are susceptible, the average number of contacts made with from a person to the infection on day T minus M. And you can see that there's one of those for each of those days between little m and big M. Now, obviously, we can't model this directly because we've got lots of these Fs and they're incredibly highly correlated. In some settings, we might expect them all to be identical. So we're going to just fit a quadratic approximation to that function. So we're going to say that for those days from minus m, little m to big M, that we can model or approximate that function by a quadratic model. So let's think if we've got this function here and perhaps we've got a big wave of COVID, so the prevalence goes up and then down and then plateaus for a little bit, and then gradually we're coming into a new wave. So if we've got this block of time that we're interested in, so our T there, we want to predict risk from then on, we're going to fit the best quadratic that we can to the period of time from little m to big M days backwards. And that gives us, this is a great graph now. So we've got our 3 quadratic coefficients that sort of approximate that curve. And similarly, we can do that at each point in time over the period of time we're interested in. So now we've got a function that's a quadratic function with these 3 numbers, B, C, and D, that are functions of time estimated from the data, and otherwise a deterministic function of the number of days back. Hopefully that's clear. All right. So we've got this hazard of dying from COVID on day T. So it's our constant term, our patient characteristic contributions, and then this linear combination of those burdens of infection over the past capital M days. And rewriting that in terms of this sort of quadratic approximation, that's approximately equal to this formula down here. And then I've just put it on the log scale at the bottom. Now, I'm just gonna, that last term, I'm just going to use an approximation again to simplify it. And also notice that this B actually turns out to be the burden of infection itself. So we can write the log of that hazard of dying from COVID-19 on day T as some constant term, a linear combination of patient characteristics, and then this term is the log of that burden of infection, and then some sort of quadratic term at the end, which essentially captures how the burden of infection has changed over the past capital M days. Right, so under a number of assumptions, we've written that hazard in this form. And one thing to notice is that it separates the patient effects from the effects of how that burden of infection is changing over time. Another thing to notice is that it is a Cox model. So the hazard can be written as a proportional hazards model. So we can expect that if we don't try and model the way in which that baseline risk is varying over time, that we will still get good estimates of those alphas and perhaps even better estimates. So we expect our discrimination to be perfectly good with a Cox model. However, those estimates of the baseline hazard at a particular time are likely to only be transportable if we're transporting between periods of time where that burden of infection is similar. So we looked at these different approaches using the data held within the OpenSAFELY platform. So we've got primary care electronic health records managed by the TPB, linked with the ONS mortality data. And this was about 17 million adults. And our outcome was COVID-19 related death, taken from data from the Office for National Statistics linked to these data. And this was early on in the pandemic, so pre any sort of vaccine, from the 1st of March 2020, right to the beginning there, for the first 100 days. And we had potential predictors were typical of the sorts of predictors that we have available to us in electronic health records. So I'll just quickly go through some of those. So demographic, we've got age, obviously, which we modeled as a spline, sex, ethnicity, obesity, smoking, deprivation, and then some characteristics of the household, rural, urban indicator, the number of adults and children, those sorts of things. A number of cardiovascular conditions are available in electronic health record data, including diagnosed hypertension, blood pressure, things like that. Some respiratory conditions, common neurological conditions, importantly, kidney and liver disease. And as we've seen in that variable selection diagram, that turns out to be quite important. And cancer, we split into hematological and non-hematological malignancies because the sorts of treatments associated with those are relevant for effects on the immune system and immunosuppression. Common immunosuppression conditions, frailty, and a few other things. So that's just to give you a flavour of the sorts of things we looked at. And how did we measure this burden of infection? Well, if you want to incorporate these measures directly, then we need a measure of it. Obviously, though, that doesn't exist. So what we started with was measures taken from mathematical models. Now, this is somewhat circular because estimates of the force of infection is more or less exactly what we want, but those estimates are derived from data that are more or less the data that we're trying to model the outcome of. So realistically speaking, it's a fairly good measure of what we want to put into the models, but it's not a measure that we could ever have in practice. So it's kind of going to be a best case scenario. And we took those for each region of England and by age group. And these account for the prevalence of infection, the way in which people interact with each other, and the proportion of the population who are currently susceptible. So it's more or less exactly that definition of F that we want. So what can we measure if we want to account for the burden of infection in routinely collected health data? Well, what we do have is some proxy measures that are likely to be correlated with that. So we have COVID-19 related emergency and accident and emergency attendances. So we counted those in the local geographical area. STP is a sort of small region. And we smoothed that over the last week. Now, A&E attendances are going to lag behind infection rate. So it's not quite what we want, and it might not be a very good measure of it, but it might be good enough. A measure that's likely to be closer to the timing that we want is COVID-19 cases in primary care. And again, we were able to get that by local geographical area and smooth it over the last 7 days. It's probably going to lag a little behind true infection rate, because obviously you have to have time to get sick and then think, oh, I want to go see my GP. And also, concerningly, the way that people interact with their GP for a disease that's emerging like COVID is likely to change over time. So the extent to which a person is going to go see their GP for COVID in the beginning of the pandemic won't be the same as the way in which they'll react to that a year later. So it's not likely to be a stable relationship. All right, so we want to predict short-term COVID-19 related mortality. So we're looking at 28 days. So we took two approaches. The first was just a classic Cox model in a cohort setting. Because it was a large sample, we took a case cohort sampling approach to reduce computational intensity. But otherwise, we can just think about this as being a classic Cox model in the whole cohort. The second approach we took was a landmarking approach, where we directly incorporated these proxy measures of the burden of infection at the start of each period. So if we just have a look at this schematic of what we did. So this is our 100-day study period, and we chopped it up into 28-day blocks that were overlapping. And at the start of each of those blocks, we took our measure of the proxy of the burden of infection and our measure of the patient characteristics for each individual, and we just essentially fit a risk prediction model in that little sub-study. And we stacked all those sub-studies to make one great big data set, and then modeled the risk within that. I hope that's all vaguely clear. And the measures of model performance that we focused on were discrimination. So as I said, this is the ability to distinguish between cases and non-cases. And there's various measures of that, but I'm going to talk about the C statistic. And calibration, which is really the focus of this. Do the observed outcomes and predictions agree? So can we model the absolute risk? And we looked at various measures there. Now, I thought that I would be coming to the end of my time here, so I haven't actually put very many of the results here, but this slide shows you the key ones. So if we just look at the bottom right, all models, whether they be the Cox model or the landmarking models, all the models had basically the same discrimination. They were all in that range, 0.92 to 0.94. So whether or not we tried to directly model that changing baseline rate didn't affect the ranking of the patients. The graph we're looking at shows us results about the absolute risk. So it's actually the risk of 28-day COVID-19 mortality predicted by the different models. And these are 3 different periods we tried to do that prediction in. So we took a period of very low prevalence of COVID, a period with very high prevalence in the middle, and then a period in the middle. And we looked at the different abilities of those models to predict absolute risks. And sorry for not putting confidence intervals around them, but let's ignore that for the moment. So on the left-hand side, the period with low prevalence happened to be at the start of our 100-day period. So our Cox model gives us a perfect prediction. It's spot on. And that's in the red dot. If we look at the middle block in the high prevalence, the Cox model doesn't know that when we move to a different time period, that underlying baseline risk has changed. So we're still predicting the same thing. And the prevalence is multiplied by 8 or so. So it's far off. And then similarly, moderate prevalence, we don't get a good estimate. The key question is, can we get a good estimate using the direct modeling? And the answer is very middling, quite frankly. So if we use those modeled estimates, which are really good indications of the burden of infection over time, but realistically wouldn't be available to us, we do get quite accurate estimates of that absolute risk, which are the horizontal lines. So we're more or less picking up the changes in absolute risk over time. However, as I said, it's not realistic to think that we could use those measures to do forward prediction in a setting like COVID. The sorts of measures we have available to us are the ones shown in yellow and purple. So the yellow being the A&E and the purple being those primary care data. And as you can see, when we go to much less accurate measures of that burden of infection, particularly those that lag behind, we're getting much less good estimates of the absolute risk and how that varies over time. So broadly speaking, what did we conclude here? Well, when we fit models that don't take account of how the baseline hazard changes over time explicitly, we get very good discrimination. However, we get typically poor calibration. So absolute risks are not transportable over time. I mean, it's not a surprise. But broadly speaking, it's very reassuring that that is fine for ordering patients in terms of their risk. However, OpenSAFELY is soon to be publishing some work looking at how those risks have changed over waves. And of course, a lot is changing about the current situation. So vaccines have come in, we've got better treatments, including those antivirals and NMABs. And that has had quite strong impacts on that ordering of patient risk. So although Cox models do a fine job at ordering patients, it is necessary to regularly update those models and not just the constant, not just the baseline hazards. If we do want short-term estimates of absolute risk for individual patients, then we do need to incorporate that estimated sort of burden of infection in the model. If we get a really good measure of that, we can get good estimates. And then that's needed for various things like figuring out the numbers needed to vaccinate and things. However, the sorts of measures that we are able to get within routinely collected health data don't perform as well, and whether those estimates are sufficiently accurate to be used, I think, is a bit of a moot point. Right. So broadly speaking, electronic health record data have a wealth of information, and there's massive and very timely resource to describe who's experiencing high risk of routinely recorded events like death from COVID. However, hopefully, as I've persuaded you, there are significant problems in using these data. We might not be able to identify the population that we're interested in. We might not have the data on the predictors that we're interested in. And we've got this data dependent sampling, which can cause all sorts of unexpected distortions in our results. And statistical approaches that we use for risk prediction aren't necessarily well aligned with what the policymakers actually want. So the sorts of answers statisticians can provide easily are not necessarily the sorts of answers that policymakers want to implement. So I do think there's a case for better communication between policymakers and statisticians and other analysts to sort of try and target the actual thing that policymakers want to implement. So I think that, in between this pandemic and the next, maybe that's something to consider. Now, I feel I must have been talking really fast because I thought that was going to take most of the hour, but that is everything I was intending to say. So just to end by acknowledging the very large number of patients whose data contributed to all of the work that the OpenSAFELY Collaborative has been doing, and the absolutely humongous team of people who've contributed. All right, I'll stop sharing, Sean, and hand over to you.
Thank you very much, Fiz. Yeah, no need to apologise for finishing earlier than you thought. The main thing is to have 20 slides or more. Right, I'll have a look. So please ask questions. I'll have a look at raise your hand. Yes, we have one hand raised. If you can't raise your hand, just type something like I have a question in the chat, and I'll call upon you. So Amani, would you, I'm sorry if I've mispronounced your name, would you like to ask your question?
Thank you, Sean, and thank you, Liz. This is fascinating, and I apologize for the noise. I have a child at home that I have slave labored into, opening his room, and I don't want to stop him doing it. I'll just say I have two questions, Liz, for you. My name is Amani Siam. I'm working with the Health Workforce Department in WHO, and we looked at a study, we did a working paper to look at deaths due to COVID amongst health and care workers. Who we mean by that are just the high risk health workers and the social workers who are, the crossover from what we call the direct care social workers. And the difficulty we've seen is that given the surveillance system in WHO, we do not see data that pertains to electronic records at all. We get aggregates. So I was just curious to know whether the data set you're working with specifies anything about the exposure or the higher risk of exposure for certain patients given the type of occupation they have. So that's my first question. And then I like very much what you've shown about the discriminant, more or less, power that you've shown in the difference between the four types of analysis. But I'm just curious about the periods. When you say a low period, a low prevalence period, a medium prevalence, and a moderate prevalence, and a high prevalence, how did you scale that or benchmark that in terms of time? Because for us, let me tell you what we observed with the surveillance data. It was very calm a bit at the beginning between March and maybe around June of 2020. And then it just poof between July and December. So my curiosity rests with whether you've picked up on that. And last but not least, did you see from these patient records any indication of the vaccination status? That's something that we're also trying to put, some sense to. Thank you very much, Liz. Back to you.
Thanks for your questions. Right. So I might just go in reverse because I can remember them better that way. So in terms of vaccination status, the data I'm talking about was just our wave 1, which was pre-vaccination. And we haven't updated this analysis because in the UK, a big working group was assembled across the UK to develop one risk prediction score for the nation in a way. And that was Q-COVID, and that sort of took over the mantle of risk prediction. And so they have done very similar work to this, but that's been updated over time. So I suggest you look at Q-COVID 3 to see what they found about vaccination, but certainly that work has been done, yeah. In terms of occupation, yes, so our data, and so the validation periods I talked about was just within the UK first wave. So we had a wave that obviously went up and down, obviously. And yeah, we just took the split into thirds, and I've just described that as low, high and medium as a sort of descriptor because that was the shape that happened, but yeah. In terms of exposure, I think that's one of the biggest flaws of using electronic health record to predict death among the general population for COVID, because we simply don't have good measures of people's occupation. And so some of these people will be at enormously high risk due to their occupation and others not so much, and we can't get a handle on that. And in the early days, we did look at trying to link to relevant data, but certain linkages are more complex than others, and it didn't happen in time.
Thanks, that's very helpful. Thanks, Liz.
Okay, thank you. Caroline? If you could unmute.
Hi, can you hear me okay?
Yes.
Oh, sorry, my camera seems to not be working. Yeah, thanks, really interesting talk. I was just picking up on the point at your later slides a little bit about communications to policymakers. I ask this because I used to work on, it was called the REACT study, and then the person I worked with on that study has now moved over to a senior role in UK HSA, and I went to a talk about that this week, and it's a lot about, yeah, how do you answer the right questions to make impact? So I was really focusing in on, you know, are you looped in with the right people, maybe at UKHSA, or do you feel that as researchers, maybe we need to pose our questions a little bit differently to get to the heart of what can make a difference from a policy perspective?
Yeah, I do think that there's a lot of questions that sort of analysts and statisticians can be asking themselves while we're sort of between pandemics. Hopefully not, hopefully it's just over now. But, you know, I think that it's a good time to be talking to policymakers about what could have been done better, and, you know, what prevented you from using this data optimally that we provided you with, and what format could we have given it to you that would have allowed you to make clearer and better decisions. And I think some of that relies on sort of very clear communication about what our models are and aren't able to do. So, I mean, we're sort of engaging in those sorts of discussions with various organisations, and I think others are too. But yeah, I think definitely more understanding of each other's perspectives is a good idea.
Okay, thanks. I guess I was familiar a bit with maybe like the SPI-M route in and stuff, and that's why, because do you think it's better to maybe have the intermediate party rather than trying to go to them and say, what data would you like? Like, that sounds like it's too much. I mean, we were having to produce the most basic bar charts to even get it across the table. So yeah.
I mean, I think my approach is to sort of talk to anyone who will listen to me.
Yeah, best way. Thank you.
Right, anyone else to
There's
ask?
a question in the chat.
Is there? Oh yes, so there is. Okay, Spencer, please go ahead.
Yeah, sorry, I'm trying to get my, yeah, I was just from like a data management side. I was wondering if sort of the dummy data that you use is a good representation of the real data that you end up using later, and if there's any like sort of, because obviously EMR data is quite messy, and I wasn't sure if like maybe the dummy data, there weren't really cleaning issues that came up during the dummy data stage that did come up later on, and if you think that there's ways that this can be improved to kind of make the analysis faster, or you know, something like that. So
yeah, and a bit more robust. Yeah, it's certainly, it's of ongoing conversation, because the people who initially designed the system are software engineers and were entirely focused on patient privacy, and so their aim was to make sure they look nothing like the real data. And of course, when I used the dummy data to implement the study design of that, the landmarking, that sort of extracted everyone who died from COVID-19 within a little period of time, and then 10 times that number of people who didn't. And of course, in the dummy data, that number of people was, you know, hundreds of thousands, and not the 6 that you'd expect in real life. And it completely messed up everything that I was trying to achieve. So having data that is nothing like the realistic data obviously doesn't suit all purposes. You know, it allows you to sort of create a few regression models, but there are problems. So the team has devised a number of solutions. So there are a number of sort of problem-specific ways of generating dummy data that have various relationships built in, and you can specify different sort of proportions, prevalences, and that sort of thing, and some correlations and some causal relationships. But yeah, there is still this sort of tension between the data not looking so like the real data that there are privacy concerns, versus having data that is sufficiently similar that it's a good test of your code. So I think it's ongoing thought.
thanks.
Right, there don't seem to be any other questions as far as I can see. I was wondering this. You model the baseline hazard, And so you're using a fully parametric proportional hazards model. And I wondered whether you assume the same baseline hazard for the whole country, or whether you allowed that to vary from one part of the country to another. Because if you don't, then I can imagine that you will, well, it'll be a misspecified model, and you'll find that some conditions appear to be be associated with more risk. Yeah, cause more risk, if you like, and they're not causing more risk.
Yes. So initially I thought it would be quite important to make sure that we accounted for local geographic area, because the patterns are all very similar, but sort of occur at different times. Like you get slightly different peaks in Lancaster versus Cornwall, and slightly different magnitudes of peaks and stuff. So we did compare Cox models with and without stratification by region, and with and without adjustment. And oddly, it actually didn't make much difference to the discrimination. But in terms of the landmarking one, because all of the measures of burden of infection were for the local area, that we assumed that was the only reason that risk would be different in the different geographical areas.
I got lost a little bit at one point, because you did indeed talk about measures that you were of baseline hazard or risk that you were getting from outside the data. And I wondered why you needed that. Was it to prevent you having to extrapolate your estimate, your quadratic assumption on the baseline hazard that you could estimate from the data?
It was really because what measures of the burden of infection that you can expect to get from electronic health record data are going to be a bit muddy, a bit poorly measured, not very good, basically. And so we wanted to be able to compare the available but not very good measures with a very good measure, and the only very good measure there was external. So it was
why do you need a measure at all? Why can't you just, you're using a parametric model. The parametric model gives you an estimate of the baseline hazard from the data. Oh
yes, it's for forward prediction. So you can only hope to predict beyond that 100-day period if you have a model that doesn't rely on being able to estimate, that doesn't rely on sort of non-parametric estimates of that particular time period. Sorry,
yeah, you don't want to extrapolate that. That's it. You are estimating a baseline hazard during those 100 days as a quadratic form. You don't want to extrapolate the quadratic form into the, yeah, I understand. Right, that's clear. Thank you. Okay, any other questions? If not, then I say thank you very much indeed, Fizz, for a very interesting talk. You haven't gone to the full hour, so you get a few points deducted for that. But otherwise, thank you very much. Very good talk. Okay, goodbye, everyone.
Bye-bye. Thank you.